All cells of the body need a continual supply of oxygen for life processes. As cells use oxygen, carbon dioxide, a waste product, is generated. Each breath brings in fresh oxygen and expels carbon dioxide. With each breath, the respiratory passages moisten, warm, and filter the inspired air, which facilitates exchange of respiratory gases. In addition to exchange of gases, the respiratory system performs many other vital functions, such as regulation of blood pH, vocalization, defense against inhaled foreign matter, and smell. Respiration is a multi-step process. Pulmonary ventilation, or breathing, is the movement of air into and out of the lungs. Exchange of gases occurs between air sacs of the lungs and blood in a process called external respiration. Transport of the blood gases, oxygen and carbon dioxide, is the job of the cardiovascular system. In the tissues, internal respiration involves exchange of gases between the blood and the cells. In this presentation on the respiratory system, we review the structure and function of the respiratory passages and lungs. The respiratory system consists of passageways that function to cleanse, warm, and humidify air as it passes down to the delicate lung tissue. These passageways include the nose, pharynx, larynx, trachea, and bronchi, which repeatedly divide into smaller branches called bronchioles. The very smallest bronchioles are the respiratory bronchioles, which transport air to the alveolar ducts that lead to the alveoli, the air sacs of the lungs. The nose consists of an external bony and cartilaginous framework surrounding an internal cavity. The bony area of the external nose is formed by the nasal and maxillary bones. The flexible portion of the nose is formed by the lateral cartilage, lesser alar cartilages, and greater alar cartilage plates. Dense fibrous connective tissue also contributes to the lateral part of the external nose. Internally, the nose is divided into two chambers by the nasal septum. The bony posterior part of the septum is formed by the perpendicular plate of the ethmoid bone and the vomer. The septal cartilage, a midline plate of hyaline cartilage, forms the anterior part of the septum. When the nasal septum is removed, the anatomy of one of the chambers of the nasal cavity can be reviewed. The opening through which air enters the cavity is the external nares or nostril. Lying superior to the nares and anterior to the nasal cavity is the vestibule, which contains hairs that filter air as it enters the nasal cavity. The roof of the cavity is formed by the sphenoid and ethmoid bones. The olfactory mucosa, containing receptors for smell, lines the upper nasal cavity. The axons of these receptor cells pass through the skull via openings in the ethmoid bone called the cribriform plate. The palate forms the floor of the nasal cavity and consists of bony and muscular portions. The anterior portion, the hard palate, is formed by parts of the maxillary and palatine bones. The soft palate is a muscular structure whose most posterior portion is the uvula. The lateral walls of each nasal cavity contain shelf-like projections of bone called conchae, which increase the surface area over which the air circulates and is conditioned. The superior concha and middle concha are parts of the ethmoid bone, and the inferior concha is a separate bone. The grooves lying between the conchae are the superior meatus, middle meatus, and inferior meatus. The respiratory mucosa, which lines most of the nasal cavity, helps condition the inspired air. It is composed of pseudostratified ciliated columnar epithelium overlying connective tissue. Goblet cells are specialized cells of the epithelium which produce mucus that traps dirt and other foreign particles entering the nasal cavity. Ciliary movement on the surface of the epithelium sweeps the mucus toward the posterior part of the nasal cavity where it is swallowed. A complex system of thin-walled blood vessels lying deep to the epithelial layer warm the air as it circulates across the mucosa. Several spaces in the skull called sinuses contain mucous membranes that are continuous with the respiratory mucosa of the nose. These paranasal sinuses also warm and moisten the inspired air and are named for the bones of the skull in which they reside. The paired maxillary bones contain two of these sinuses. Ethmoid air cells within the ethmoid bone contribute to the ethmoid sinuses. Two other sinuses, the frontal sinus and the sphenoid sinus, are seen in this view of the head. All of these sinuses drain mucus through openings into the nasal cavity. The pharynx, a skeletal muscular tube lined by epithelium, transports air and food and is divided into three regions by several anatomical landmarks. The nasopharynx lies posterior to the nasal cavity and extends to the level of the soft palate. The oropharynx is the portion of the pharynx which lies behind the oral cavity between the soft palate and the hyoid bone. The laryngopharynx extends from the level of the hyoid bone and divides into the esophagus posteriorly and the larynx anteriorly. Air leaves the nasal cavity through each internal nares and enters the nasopharynx. Usually, only air is transported through this portion of the pharynx. The lining of the nasopharynx is a continuation of the pseudostratified ciliated columnar epithelium that lines the nasal cavity. Both auditory tubes open from the middle ear cavities into the nasopharynx. One of these openings is seen in this specimen. The auditory tubes help to equalize air pressure in the ears with the atmospheric pressure of the nose and throat. Paired collections of lymphatic tissue called pharyngeal tonsils, or adenoids, are located on the posterior wall of the nasopharynx. Located in the oropharynx are the palatine and lingual tonsils. The laryngopharynx terminates by splitting into the esophagus, which transports food and drink posteriorly, and the larynx, which conducts air toward the lungs anteriorly. The oropharynx and the laryngopharynx transport both air and food. They are lined by non-keratinized stratified squamous epithelium, which is more protective than the respiratory epithelium of the nasal cavity and nasopharynx. In this specimen, the anterior chest wall and several of the blood vessels and other soft tissues in the neck region have been removed so that we can see the location of the larynx and trachea. The thyroid gland is the large structure which surrounds the larynx and upper part of the trachea. 9 separate cartilages supported by muscles and ligaments form the walls of the larynx and connect the laryngopharynx with the trachea. The cartilaginous structure of the larynx helps to assure that the tube conducting air to and from the lungs remains open. The larynx is anchored to the hyoid bone by the thyrohyoid membrane. The unpaired thyroid cartilage is the largest of the 9 cartilages that form the larynx. Its laryngeal prominence, sometimes called the Adam's apple, is usually larger in males than in females. Attached to the thyroid cartilage by the cricothyroid ligament is another unpaired cartilage, the ring-shaped cricoid cartilage. This cartilage can be seen more easily in a posterior view of the larynx. A triangularly shaped pair of cartilages, called arytenoid cartilages, are located on the superior part of the cricoid cartilage. These cartilages attach to the vocal cords and are very important in vocalization. The paired corniculate cartilages are situated on the arytenoid cartilages. Another pair of small cartilages, the cuneiform cartilages, lie anterior to the corniculate cartilages in the aryepiglottic folds attached between the arytenoid and epiglottis cartilages. The epiglottis, a large, flexible, unpaired cartilage, attaches to the anterior rim of the thyroid cartilage. When the larynx is viewed from above, shelf-like structures of tissue extend into the laryngeal opening from its walls. These are the vestibular folds or false vocal cords. The vocal folds, or true vocal cords, lie inferior to the vestibular folds and are formed from 2 bands of elastic tissue ligaments stretching across the opening of the larynx. The glottis is the medial opening between the vocal folds. When air rushes by, the vibration of the vocal folds results in the many sounds of speech. The vestibular folds have little or no involvement in vocalization. The vestibular fold and vocal fold are also visible in this mid-sagittal view of the larynx. The space located between the folds is the ventricle. The epithelial lining above the vocal fold is a continuation of the non-keratinized stratified squamous found in the laryngopharynx. Below the vocal fold, the epithelium changes to pseudostratified ciliated columnar epithelium, which continues to line the tracheal portion of the respiratory system. The trachea is the inferior continuation of the larynx. When the thyroid gland and the aorta and its branches are reflected, we can see its pathway through the chest. The trachea is about 4 to 6 inches long. The trachea branches at the carina into the right primary bronchus and the left primary bronchus. Notice that the right primary bronchus is shorter, wider, and more vertical than the left primary bronchus. This is why inhaled objects are more commonly lodged in the right bronchus than in the left bronchus. There are 16 to 20 incomplete cartilage rings in the trachea. Posteriorly, each incomplete ring of cartilage is completed by the trachealis muscle. Pseudostratified ciliated columnar epithelium lines the inside of the tube. The lungs are protected by the ribs and are located within the pleural cavities. They are surrounded by a double-walled serous sac, the pleura, which was cut when the thorax was opened. The outer layer of the pleural sac, the parietal pleura, tightly adheres to the muscles of the thorax and to the diaphragm. The inner layer of the pleural sac, the visceral pleura, lies directly on the surface of the lungs. The space between the parietal and visceral pleura contains pleural fluid which lubricates the surfaces and reduces friction between them during breathing movements. In the living state, this space is very small. As can be seen in this specimen, the right and left lungs are not the same size. The left lung is smaller because of the location of the heart within the thorax. The cardiac notch on the surface of the left lung marks the impression of the heart. We will now examine a lung preparation which has been removed from our specimen. The apex is the superior pole of each lung. The diaphragmatic surface is the arched base of each lung, which is shaped by the diaphragm. The costal surface is the lung area surrounded by the ribs. The mediastinal surface is the portion of the lung which surrounds the heart and contains the structures which enter and exit the lung. The surface of the lungs are subdivided into lobes by deep fissures. The right lung, seen in this right lateral view, is larger in mass and divided into 3 lobes by 2 fissures. The superior lobe is separated from the middle lobe by the horizontal fissure. The oblique fissure separates the middle lobe and the inferior lobe. From the left lateral view, we can identify the 2 lobes of the left lung. The superior lobe and the inferior lobe, which are separated by the oblique fissure. The lobes of the lungs are further divided into regions called bronchopulmonary segments. Each lung contains 10 bronchopulmonary segments. Connective tissue separates these segments from one another. In addition, each bronchopulmonary segment has its own vascular and airway supply. Because of this compartmentalization of the lung tissue, disease is often restricted to one or just a few of the segments without spreading to other areas of the lung. The bronchopulmonary segments are further divided into smaller lobules. The many branches of the respiratory passages are collectively referred to as the bronchial tree. The bronchial tree is functionally divided into conducting and respiratory zones. The respiratory passages that comprise the conducting zone cleanse, warm, and humidify air as it passes down to the delicate lung tissue. The respiratory zones consist of terminal portions of the respiratory passages that participate in gas exchange. We will review the passageways which constitute the conducting zone first. As we reviewed on our specimen, the right and left primary bronchi, the first part of the bronchial tree, are formed when the trachea divides. The primary bronchi continue to divide to form the secondary or lobar bronchi. Each secondary bronchus supplies a lobe of the lung, 3 on the right and 2 on the left. The secondary bronchi divide into tertiary or segmental bronchi, which divide again and again, with each division resulting in a smaller diameter branch. Overall, there are about 23 orders of branching in the lung. When the diameter of the air passageway is less than 1 millimeter, the passageway is referred to as a bronchiole. The smallest and terminal branches of the bronchioles are less than half a millimeter in diameter and are called terminal bronchioles. It is important to remember that no gas exchange occurs in the air passageways discussed up to this point. We will now examine the passageways which constitute the respiratory zone. The terminal bronchioles empty into the respiratory bronchioles, which are the beginning of the respiratory zone. The outpocketing of alveoli from the walls of respiratory bronchioles distinguishes these bronchioles from terminal bronchioles. Alveolar ducts lead from the respiratory bronchioles to the alveolar sacs, the end of the air passageway. An alveolar sac consists of clusters of alveoli, much like grapes at the end of a vine. Accompanying the terminal bronchioles into the respiratory zone are a pulmonary arteriole and a pulmonary venule. These vessels contribute to the formation of the pulmonary capillary bed, which blankets the alveoli. The lung contains an astonishing number of about 300 million alveoli, each about 0.3 of a millimeter in diameter, which accounts for the huge surface area available for gas exchange. The thin wall of the alveolus is composed of a single layer of simple squamous epithelium supported on a basement membrane containing elastic fibers. These simple squamous epithelial cells are referred to as type 1 alveolar cells. Also scattered along the wall of the alveolus are type 2 alveolar cells, which secrete surfactant to decrease surface tension within the alveolus. Alveolar macrophages, sometimes called dust cells, wander throughout the alveoli, phagocytosing foreign particles in the lungs. The wall of the blood capillary is formed by a simple squamous epithelial cell called an endothelial cell, which lies on a thin basement membrane. The respiratory membrane is very thin, only about half a millimeter thick. The simple squamous epithelium of the alveolus and its basement membrane, together with the simple squamous epithelium of the capillary and its basement membrane, constitute the alveolar capillary respiratory membrane. This air-blood barrier separates the gas in the lungs from the blood circulating through the pulmonary capillaries. Exchange of respiratory gases occurs by simple diffusion. The inspired oxygen moves from the alveolus into the pulmonary blood. The circulatory system distributes the oxygen-rich blood throughout the body. Carbon dioxide moves in the opposite direction from the blood into the alveolus, where it will be expired from the body. Normally, gas exchange across the respiratory membrane is rapid due to the extreme thinness of the respiratory membrane. However, anything that thickens the respiratory membrane impairs gas exchange. For example, in pulmonary edema, the interstitial space becomes swollen with fluid. As the distance between the alveoli and pulmonary capillaries increases, the rate of gas movement across the respiratory membrane slows. Consequently, as the blood moves through the pulmonary capillaries, there may not be sufficient time for adequate gas exchange. In this presentation, we have reviewed the structure and function of the respiratory system and the passageways through which air travels to and from the lungs. Gas exchange across the respiratory membrane in the lungs was also illustrated. When food is ingested, it is composed of many large molecules which contain nutrients that give us energy for all bodily activities. However, the food that we ingest must be broken down into smaller molecules and absorbed into the blood before it's useful as an energy source. These activities are the functions of the organs of the digestive system, which includes the gastrointestinal tract and its accessory structures. The gastrointestinal tract, or GI tract, is essentially a long tube open to the environment at both ends. Food enters the GI tract through the mouth. Mastication by the teeth begins the process of mechanical digestion. Secretions from salivary glands contain enzymes to begin chemical digestion and mucus to bind the food into a compact bolus. The tongue moves the slippery food bolus to the back of the throat where it is swallowed. The food bolus is propelled by peristaltic waves down the esophagus to the stomach. In the stomach, food is stored temporarily. During this time, it's mechanically broken down into smaller particles. Some chemical digestion and very limited absorption also occurs in the stomach. Food is slowly released from the stomach into the small intestine where the majority of digestion and absorption takes place. Secretions from the liver, gallbladder, and pancreas are released into the proximal part of the small intestine to aid in digestion of food. The unabsorbed foodstuffs remaining in the small intestine continue down the tract and enter the large intestine or colon. Absorption of water occurs throughout the length of the large intestine, concentrating the remnants of ingested food. This waste is referred to as fecal matter, which is excreted from the body. As you know, the mouth receives ingested food. Several digestive processes occur